Okay, can you guys, is screen sharing enabled? Varsha, are you here?
Yeah, just a minute. I
Yeah,
forgot doing it.
I'll just get my drink of hot tea so that I can keep talking. I'll put myself on mute while. Okay, so while we wait for the others, do the early birds have any thoughts from the last lecture that they wanted to talk about, clarify? Just say, wait a minute, I didn't understand that, or wouldn't this be a cool thing to study in the future? Does anyone want to have thoughts from that lecture? So what happens after your neuro section? What is after neuro? What do you guys study?
I think we'll be studying organogenesis.
Organogenesis. So like, who takes that?
I think it's Arjun who's taking it.
Okay. So more lung or I don't know. Is it one class? Many classes?
2 classes.
2 classes. Okay. So this is nice. You guys kind of get a flavor of everyone and the kinds of things that interest them. It's good. Did you guys see something on social media that's talking about Zoom and what it's like to give talks on Zoom? I'll tell you, this is so that I saw this. Are they called memes? I don't know. But there's this guy talking really passionately, and then you zoom out, and there's a brick wall right in front of him. Kind of feels like that, brick walls made up with names, lots of names. But hopefully this will lift and everyone can see each other and chat with each other. Okay, we have how many people now?
Oh yes, I've seen that meme where the guy just
Very passionate.
standing and staring at.
And there's this brick wall in front of him.
Yes.
Famous images from 2020.
Yeah.
And then did you guys see the other one that says 2020, written by Stephen King, produced by Steven Spielberg?
No, this I haven't.
I like that one too. You can't get bigger than that, huh? Okay, it's 35. Shall we dive into it, Varsha? What do you think?
Yeah, sure. I mean, it's already 5 minutes.
Yeah.
There are 20 people. No.
And people will trickle in or not, or trickle out. Who knows? Okay, so let's get started. So this is your second class on neurodevelopment. And remember, this year we've split it up into vertebrate and invertebrate. The first 2 classes will be invertebrate, which I will take, and the next 2 classes are going to be taken by Bhavana on vertebrate nervous system development. So just like last time, that's my email address up there. So please feel free to bug me with anything that interests you, sparked your curiosity, doesn't make sense, whatever. And I'm always happy to chat. Okay, let's get started. So I thought I'd start by just doing like a recap of what we did in last class. Remember, like I said, we've divided up into vertebrate and invertebrate, and do please pay attention in the next classes for any recurring themes. You'll find a lot of these recurring themes, and it'll be fun to keep that in mind as y'all are hearing more about vertebrate development. So just to recap, so the first thing that we did, and these again are themes that you will see in both vertebrates and invertebrates. First, as you develop a nervous system, you need to set aside tissue that has neurogenic potential, and we studied that in some detail in invertebrates. So that's the neurectoderm. How do you specify the neurectoderm? And in the vertebrate, that's the neural plate. So you'll see how that is done. And you'll find some very similar processes, including genes involved in this process. So once we set aside neurectoderm, we did something in which is kind of unique to flies, which is we made sure that not everything in the neurectoderm became neurogenic. We kind of restricted the progenitor fate over there. And once we restricted that, out popped these neural stem cells into the embryo, creating along the anterior posterior axis of the embryo like a sheet of neural stem cells. And this is analogous, I won't call it homologous, to the process of generating a neural tube out of the neural plate that you'll see in vertebrates. Keep that in mind, analogous, not homologous. Okay, and then so we learned that, and then this whatever neural stem cells that are spotting the embryo from the anterior to posterior, we gave it axial information. So we gave it dorsoventral information, and we gave it anterior posterior information. So we learned, this is kind of what we did in the last class. For those of you who are missing then and need a quick catch up, you can, I believe these talks are recorded, and so you can always go back and look at it, unlike in that. I guess that's an advantage of having Zoom lectures. Okay, and so now this class, what we're going to do is we're going to see how now from all of these stem cells do you like suddenly have an explosion of the numbers of cells that you need. Remember, we discussed those neurons as many as stars in the Milky Way kind of numbers, right? So how do you get the stem cells to generate all of those different kinds of neurons and glial cells, not to forget glial cells. And then we're going to talk about temporal patterning. So this is very special, and we'll get into it later. So again, just one slide recap of each one of those things. We first started off by specifying the neurectoderm. Remember, in vertebrates, this is happening ventrally, specified. And sorry, did I press something wrong? No. The thing that I love about this, and I want you to take note of, is that all of this patterning that we have studied, the embryo is actually, the nervous system is using, it's piggybacking on everything that the embryo is doing. So the embryo does nothing special to impart axial information to the nervous system. It's using the same dorsoventral patterning genes to pattern the nervous system as it did to pattern the entire embryo. So here's your dorsoventral patterning gene, the morphogen dorsal, which the mom has put into the embryo. And that region is specifying, that expression is specifying snail. Remember, in the ventral most region, and any tissue that's expressing snail becomes mesoderm. It gets invaginated through gastrulation and forms the mesoderm. And those green bits at the end, which also dorsal is specifying, is short gastrulation. And that, because of gastrulation, kind of comes together and becomes the ventral neurectoderm. And that is what is, you've now specified the neurogenic tissue. Okay, then we learned how from there you need to now make sure that you're restricting fate. So you've got these little clusters over there, which are you've made proneural clusters. And then from the proneural clusters, you use this beautiful system of lateral inhibition through notch-mediated signaling, such that from each one of those clusters, you pick only one neural stem cell. And then after that is when you remember that this is happening in each one of these clusters, and all of these neural stem cells along the entire length of the embryo popped into the embryo, and you get this structure that we're calling analogous to the neural tube. Okay, then what did we learn? We learned that this thing needs to be given information about where it is, and we learned how that same dorsal gene at different concentrations is giving you VND, IND, and MSH. And then we learned, so that's your dorsoventral axis. And then we learned that the same anterior posterior axis information is giving anterior posterior axis information to the neural stem cells. And then on top of that, so that is per segment. And then on top of that is the homeodomain containing transcription factors, which are really giving you you are head, you are tail, you are in the middle. And that at the end of all of that, we learned how you get this segmentally repeating unit of neural stem cells that are molecularly distinct from each other because they all experience unique AP and DV patterning genes. And what this meant was that you could end up with each neural stem cell now being able to give rise to different types of neuronal lineages, different types of neurons out of each one. So that's the recap. And now we are ready to talk about cell division. So now that you have, so we saw that the neural stem cells that are different from one another giving different lineages, but even that in that there are many, many different neurons and many, many different types of neurons. So we're going to talk about how do you get the many, many different neurons and many, many different types of neurons. That's the topic of this class. So cell division strategies. So let's start with that. So that orange blob over there is the neural stem cell. In invertebrates, that's often called the neuroblast. So you'll hear me using these words interchangeably. So there are a few different kinds of strategies that are used by neural stem cells to give rise to all of the different neurons. And there are three that are prominent, and we'll discuss what those three are. The main thing to keep in mind is that the neuroblast needs to divide and create that bunch of neurons. And what we need to make sure of is that cell number. It needs to create the appropriate cell number. So what cell division strategies would it use to give rise to the number of cells that particular uniquely defined neural stem cell needs to give rise to? So here's the first strategy. So a neural stem cell can divide, and remember it needs to keep dividing throughout life, right? So it undergoes what's called asymmetric cell division. Have you guys encountered this in your Dev Bio class yet? The idea of asymmetry, that when you divide, there are different ways of doing this, right? When you divide, the two progeny can be exactly the same as the mom, or the two progeny you can make different. And in this case, what you're doing is you have to make things different. These are stem cells. So the reason you need to make them different is because no matter what, you need to retain that stem cell potential. It needs to be able to go on and keep giving rise to neurons or wherever the tissue. If you're looking at, I don't know, stem cells in the intestine, it needs to keep generating intestinal tissue. But if you're in the brain, you need to keep generating neurons. So in this type of asymmetric cell division, each asymmetric cell division is giving rise to one postmitotic neuron. And so this stem cell can now keep ticking away, and with every division, it can pop out one neuron, right? And at the end of the development, this particular neural stem cell undergoing this pattern of cell division will give rise to, say, let's assume it divided 4 times. It'll give rise to 4 neurons, right? This type of cell division is called type 0. Okay, and you'll encounter this when Bhavna talks about this. There's something very similar, this strategy of cell division, very similar in vertebrates, and it's called direct neurogenesis in vertebrates. Okay, so make a note of the cell division, the cell number potential that this type 0 type of division gives rise to. Let's discuss another one. So you could have cell division. Again, it has to be asymmetric because you need to self-renew. But in this case, you don't pop out a differentiated neuron. You pop out this intermediate type of cell called a GMC, or a ganglion mother cell. And has anyone encountered ganglion mother cells before? Okay, I'll leave that for you to think about. But ganglion, Pratiksha, are you saying something? Or you're just connecting? I'm not sure. Okay, so ganglion mother cells are not postmitotic. They have the ability to divide once more. And now when they divide symmetrically in a sense. That's not entirely true, but in terms of what they give rise to, they divide symmetrically to make two neurons, two postmitotic neurons. So now in this case, each division of the neuroblast, we're going to ignore the fact that it's via a GMC, but each division of the neuroblast is not giving rise to one, but two neurons. And this can now keep ticking away. The same, imagine it goes through the same 4 cell divisions of the neuroblast that we saw in the previous slide. But now what do you end up with? Twice the number of neurons, right? So asymmetric cell division, same neuroblast, asymmetric cell division, but because you generated this intermediate type of cell, you now have the ability to double your cell number potential. And this type of cell division is called type 1. And it's called that because for the longest time, this was the only one that we knew of in the fly brain. And so it was called type, and it didn't even have a name then, it was just called cell division in the brain. And then as people started discovering other ones, this one began to be called type 1. Now again, and actually it's the most abundant type of cell division that you'll see in the fly brain. And this too has a vertebrate homologue. And in the vertebrate, this is called indirect neurogenesis because you have that, that intermediate GMC type of cell. And again, you'll hear about this from Bhavna. Okay, so that's 2. We've discussed 2 strategies. There's a third strategy, which is really important in the fly brain. And in this, you do something even more funky. You take that neuroblast, of course, everything, asymmetric cell division, just that's, you burn that in your brain. Undergoes asymmetric cell division. So it self-renews, you have the neuroblast, and then it generates another intermediate type of cell, but this is like a superhero intermediate, and it's called an intermediate precursor, INP. Very boring name for a very neat guy. And what the INP can do is basically it masquerades as a type 1 neuroblast. And so when it divides, it undergoes asymmetric cell division, right? So now it's self-renewed itself. So it's got an INP and it generates a GMC. And now you all know what GMCs do. They divide once more to give you two postmitotic neurons. So now does anyone want to come in and tell me what might happen? Let's follow the INP. What might happen with the INP now? Does someone want to make a guess, a wild guess? So we have an INP sitting over there.
Will it make another INP and one GMC?
Exactly right. That's exactly right. It does what the type 1 would do. It divides once to make an INP and a GMC, and the GMC divides again to give rise to 2 neurons. And tuck, 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 tuck. Remember, we're following 4 divisions. So at the end of 4 divisions, you get your 8 cells, right? But now does someone want to step in and say, there's that from the first division over here, from this first division, we had 1 INP and we had the second neuroblast. That's kind of sitting around while we've dealt with what's going on with the INP. So does someone else want to step in and say what might be happening to that neuroblast now?
That would again asymmetrically divide to give you an INP and another neuroblast?
Exactly that. So now, because it's a neuroblast, it's a type 2 neuroblast, it divides asymmetrically, it gives you a neuroblast and an INP. Super boom. And now what happens, as Nivedita said, now this INP is hanging around, right? It's not going to just hang around. It's going to go through its set of divisions. So now what have we done? We have, remember, all we've dealt with are 2 neuroblast divisions. And in those 2 neuroblast divisions, we have generated how many neurons? Each one of these is giving us 8, right? So that 4 divisions that we were considering in the previous slide of the neuroblast, those 4 divisions of the neuroblast in type 2 neuroblasts is giving you 8 plus 8 plus 8 plus 8 minus a few where you have the out of syncness of the INP with the neuroblast division. So you have quadrupled the number of cells that you can make out of type 2 type of divisions. And this is called the type 2 because this was what was discovered after the type 1 divisions. And this is a beautiful way of making lots and lots of neurons. And not only that, this is what is used extensively in vertebrates to really give you those large numbers, those Milky Way scale of numbers that we heard about in the last class. And so this is also a type of indirect neurogenesis, but it uses a combination of type 1 and this special superhero intermediate precursor cell. Okay, so basically just to put all of that together, you have type 0, which gives you 1 neuron per division. You have type 1, which gives you 2 neurons per division. And then you have these type 2s, which gives you a whole bunch. And I'll tell you, so the type 1, the type 2s, the INPs actually, they don't go on infinitely like the type 1s do for all of larval life. So they'll go through a set number of divisions and then they'll stop. And so I think with each INP division, you get about 12, 24, a couple of dozen neurons at max. So you're increasing, as you can see through all of these division patterns, you're increasing the cell number potential depending on which strategy you use. Okay, any questions here?
Hey Sonia.
Yes, Akash.
Yes. So what decides that neuroblast goes into which type of division?
Interesting. So for the type 2, for example, it seems to be that decision seems to be made even as it is specified, you know, that very early stage of it popping out of the neurectoderm. It seems the deal seems to be sealed at that stage for the type 2 neuroblasts. And that's probably because of the spatial information that they're receiving and certain factors, certain types of proneural genes, which are apart from the ones that we discussed in the last lecture. So we discussed Akete-Skute and Lethalov-Skute. There are others. So there's ASENS and there's DEADPAN. And these combination of genes allow the type 2 to do what it does. And that decision seems to be getting made even as the type 2s are popping out of the neurectoderm.
Okay, so basically neuroblasts are already destined to which type of division they'll undergo.
In the invertebrate, that is certainly true. So this is something that you will see throughout as you start reading about, or you if you're interested in nervous system development and you start reading about it, you will notice this is the major difference between invertebrates and vertebrates. Invertebrate nervous systems are much more deterministic. Decisions seem to be much more genetically encoded. There's less, I'm not saying there isn't, but there's less plasticity in the system. It's much more hardwired, genetically controlled. The vertebrate nervous system, you'll see there's much more pliability. You can move things around from one location to the other and they can take on identities of that new location. So there's much more of that kind of thing. So it's true, in what you're saying is true. In invertebrates, a lot of that decision is made even as you're being born as a neural stem cell. So that's not to say that indeterminism doesn't occur in invertebrates. And I wonder now, you know, a lot of these things that get written in textbooks, it's often worthwhile going back to them and thinking about them again to say, you know, what if we were able to move a stem cell from one location? And by move, I don't mean physically move. You could do that, but you can do genetic moving effectively So what if you were to take a stem cell from here and move it into this location, or genetically change the spatial information in that location? Can you actually change what it's fated to be? So these are interesting questions.
Yes. Okay. Yeah. Thanks.
Yeah, thank you for the question. Anyone else?
Yeah.
Atal,
So,
go ahead. Is that Atal saying something or Girish saying
no,
something?
Girish.
Girish, go ahead.
Yeah. So the neurons being produced here are the larval neurons, right? So what happens in the adults? Do they save some neuroblasts or do they repurpose these neurons?
They do both. We're going to talk about that a little bit. They do both. You're absolutely right. We're talking about Drosophila. These are holometabolists. So they go from the embryo to larva, and then from larva to pupa to adult, right? So what effectively the fly needs to do is make the nervous system twice over. But it does that by not making different set of stem cells for the adult. It uses the same stem cells as it's specified all the way back in the first couple of hours of embryogenesis. And what it does is it very quickly, so the embryonic life is about just short of 24 hours. And in that much time, all of this action has to occur. You need to make the neuroectoderm, make the neural stem cells, quickly generate a whole bunch of neurons that will make a fully functional larva. And remember that larva actually has the ability to crawl, taste, smell, learn. It does all of that in search for food. It needs to munch. So it needs a fairly decent, fully functional nervous system, which these neural stem cells generate in this 24-hour period. Then at that, once embryogenesis has happened, these neural stem cells, after doing this really busy work of generating all of these neurons, kind of relax and go into what's called a quiescent phase. And after that quiescent phase, as the larva comes out and it starts eating, that nutritional cue comes into, this is again very interesting biology, which we will not cover over here, but those nutritional cues kind of reactivate these stem cells, and then it launches into the next 4 days. All of these stem cells that you saw in that previous slide start dividing again with all of their different strategies and generate all the much more complex nervous system that's required for the adult. And it's not that the larval neurons then die. Actually, what we found is that most of those neurons get repurposed. So there's dramatic rewiring of all of those neurons to wire into the adult circuitry. So both of what you're saying occur, Girish.
Okay, yeah.
Okay, and then Atal, I think you were going to say something.
Yeah. So I was just wondering, how does the last INP know that it does not have to divide into a GMC? So what kind of cell state change does it undergo?
Yeah, I don't remember exactly what happens with the INP, but people do study that last division and say, what happens? How does a neuroblast now know that it needs to stop and not generate anymore? And there are different strategies for doing this. One common way of doing it for the neuroblast, I don't remember what happens with the INP, but one common way of doing it for the neuroblast is the neuroblast will go into apoptosis. So at the end, it just dies. And what triggers that are actually your Hox genes. So it'll know there'll be a sudden pulse of a Hox gene, and ABD is a common one that really, it's been studied most in the abdominal segments. That's why I'm saying ABDB. That Hox gene, suddenly the neuroblast has this urge of ABDB, and then apoptosis occurs. So that's how a lot of termination of cell divisions occurs in the neuroblast. Another way of doing it is that the last division, this is not as common, but the last division is symmetric. And so you don't have self-renewal anymore. So those are the two ways that it's done at the neuroblast axis. The INP axis, it must be using something similar, but I don't remember what that is.
Okay.
Okay, and then
Sonia.
Yes, INP.
So this mechanism, the one that you mentioned where the last division becomes symmetric. So on that thread, I'm wondering if it's possible for the same neuroblast to switch division types. Can it go from being
Yes,
a type 1
absolutely.
to type 2?
Type 1 normally doesn't go into type 2, but you can make it. And
Okay.
so who was it who was asking about, I think Akash was saying, so you can do genetics and convert type 1 divisions into type 2 divisions. And there's a whole bunch of genes, you know, the things that I was talking about, ASENS and BRAT, and all of these things that can actually allow you to take a type 1 and send it into a type 2 division pattern. And actually, that's how type 2s were discovered. Then type 1 to type 0 is very common. And this again happens in the abdominal regions, in the segments that make the abdominal, make up the abdominal segments, basically. And you can imagine why so much of all of these strategies occur much more in abdominal segments, because it's far less complex. You need much fewer, excuse me, you need much fewer neurons in the abdominal segments. So there are all of these interesting ways of making sure you terminate neuroblast early, making sure you reduce the potential of neuroblast, cell number potential of neuroblast. So type 1 to type 0 switch is very common in neuroblasts in the abdominal segments.
Okay, thank you.
Okay. Oh, and just on that line, so the type 2s, so, you know, we spoke about how the abdominal segments have much fewer neurons. So there are ways to switch the number potential in the abdominal segment. The opposite happens in the brain, because you have this massive thing in the front, right? So most of your type 2s, all of your type 2s, there are only 8 per side in flies. And all of those 8 are in the brain. And you can imagine why, right? You need to make many more cells for the brain than you do for the ventral nerve cord. So all of the type 2 divisions are in the brain. And the other interesting thing about the type 2s is all of those 8 plus 8, 16, generate something like, and there are 100, so say 200 neuroblasts, out of which 16 are type 2s. I think it's something ridiculous, like 60% of the neurons are generated by type 2 divisions, despite being a much smaller number. So just gives you an idea of the potential of cell number that it gives rise to. Okay, that was nice to get some questions and helps me think as well. Like, for example, I've forgotten about INPs and what terminates their division. I'm going to go back and look at it. Thank you, Atal, I think. Okay, so let's move on. So that's right, sorry. So you saw in all of this, I kept on and on about asymmetric cell division. You need to do things differently in your daughters. So let's now talk about asymmetric cell division. This really lies at the heart of being able to generate all of these different numbers. So here's a picture of 2 neural stem cells dividing in the brain. And you can see just from, so this slide is just a beautiful slide. Okay, so over here you can see just from the size and shape that asymmetric cell division is occurring, right? So the big guy there is likely the neuroblast, and then I know because I've imaged this. This is a type 1 neuroblast. So I know that what you're seeing over there is probably a GNC. Okay, but these things are actually very beautiful to watch live, and I don't do that kind of work, but there are many labs. I'm sorry, I'm missing the reference for this, but there are many labs that study asymmetric cell division. And if you have an interest in cell biology, this is really beautiful. And you like imaging, this is a really beautiful field. So in this movie, which I'll play for you in a second, you'll see the neuroblast is labeled via this. We can talk about what this is, but it's a molecule that's allowing you to see. It's labeled with GFP. And so you can see the neuroblast over there. And in this channel over here, you have another gene, which is called Miranda. We'll encounter it in a second. But the authors have imaged these 2 things together, and you will now see how beautifully this neural stem cell is dividing. And there you go. So it's dividing asymmetrically. You can see that, right? So now you're not only looking at size and shape as you did over here, but what you're seeing is, look how this particular factor gets localized completely only in that GMC. Whereas this factor, maybe you didn't see it, but when you look at, I'll play the movie again, you look, this factor will get localized as a crescent up on top over there. I'll play it again for you to see. Can you see it? It got localized to the opposite end of that Miranda expression. So another movie to show you the asymmetry. And here's a movie that shows you just how, just how the neural stem cell is dividing, and it just keeps going at it. Each division takes about an hour, maybe 20 minutes, somewhere around that range. And it just keeps popping out through asymmetric cell division, all of these neurons. And if you were to open up the brain, if you weren't doing live imaging like this, if you were to open up the brain and look at it, the brain is actually full of all of this. So this is a larval brain, a third instar larval brain. You can see stem cells sitting over there with all of their progeny neurons sitting around. And these now, Akash, I think was saying, they know what they're going to be. So all of those neurons will then, through pupil life, they're being made in the larval life, but through pupil life, they really send out their axons and they make their synaptic connections. And when you open up the adult brain, you get beautiful things like this. So the pictures that I'd shown you of lineages in the last class were at the end of embryogenesis, so the larval lineages. And now I'm showing you what it looks like in the adult. Okay, so a lot of work has gone into how you get asymmetric cell division. I will briefly touch on this, but I have put in a slide with a link to 2 lectures by Jürgen Knoblisch, who really spent a lot of his early career looking at this. And they are beautiful eye biology lectures. Have you guys encountered any eye biology lectures before? If you haven't, do explore that website. It's just wonderful. They bring experts who really work in the areas that leaders in their fields, and eye biology curates them and makes very excellent teaching quality lectures. So please do explore that that platform for talks, and look at Jürgen's talk, because as they say, you must hear it from the horse's mouth. But the general idea is that when you need to get asymmetric cell division, you need to take that stem cell. And the first step, well, we're not even talking stem cell. You can talk about embryos. You can talk about C. elegans embryo is where a lot of this work has really been pioneered. The C. elegans is so good. The embryo is nice and big. It's transparent. You can image things live. The cell divisions are easy to observe, and and you can do genetic screens. So a lot of our understanding of how you break symmetry, that's the first thing you need to do to bring about asymmetric cell divisions. You need to break symmetry. So a lot of that work has happened in actually C. elegans embryos. So what does it mean to break symmetry? So you saw it in that video that I showed you of Miranda and APKC and the neural stem cell. You need to kind of segregate your proteins differently. That's breaking symmetry. And so you would maybe do something like this. Now, how do you, breaking symmetry again is one of those things that that's interesting, because as soon as you find a genetic factor and say, okay, this is localized posteriorly, and then you kind of scratch your head saying, okay, so what makes sure that that is localized posteriorly? So kind of, you keep going back. But at the heart of all of these is randomness, actually, that allows you to take a chance event that you can then amplify. We saw that with lateral inhibition as well, right? So I really like this idea that inherent noise is exploited by biology to seal deals and establish fates. So symmetry breaking is kind of a process like this. And of course, you can have influences. So for example, niches, stem cell niches are a very good way of breaking symmetry, where you have information coming in from your environment that allows you to break symmetry. But this is a beautiful, beautiful area in biology, breaking symmetry. So what have we done? So we've taken a neural stem cell, we have broken symmetry and segregated factors. And the other thing that you need to do is you need to now, after you've broken symmetry, you need to make sure that cell division occurs in the plane, in line with that axis that you have set, whatever symmetry breaking event has set up. So imagine that if you've broken symmetry in the way that we showed over here, but if you didn't orient your mitosis correctly, if you oriented it kind of like this, then all of that effort of segregating those factors in a certain way is useless, because both the progeny have all kinds of things going on. But if you actually aligned your mitosis, your cell division, to this axis of polarity that has now been established, you can then end up with a neuroblast and a GMC, right? So what are the details? As I said, there are lots of molecules. People who work on this have worked out intricate details of how this occurs. I will summarize it very briefly with this slide over here, basically because this is such an involved field that I don't know it so well. And so I'll guide you to Jürgen's talk for it. And he's done such a wonderful job with it. But the two things that we discussed, segregation of molecules, you can see that in the first circle over there, and alignment of the mitotic spindle, which is in the middle over there. So I'll just draw your attention to a few important factors. So if you look at inscrutable over here, inscrutable is one of those factors that was first identified for its ability to align the spindle. So you'll see in Jürgen's talk, he talks about, because he was one of the ones who actually demonstrated this. If you fiddle around with inscrutable genetically, you can actually change the axis of cell division. And so then, of course, you have to ask yourself, what makes sure inscrutable is localized correctly, and its partners in crime are this set of genes over here. So pins alpha, G alpha, I, and MUD, they make sure that inscrutable is pinned at that apical domain so that it can then pin the mitotic spindle where it does. And then these genes over here, the PAR3, PAR6, and APK6, remember you saw APK6 in that movie earlier, they are again apical factors. So they are actually, the PAR complex is actually one of the first things that breaks that symmetry. And they were discovered in C. elegans. And so that complex is basically allowing inscrutable via pins and MUD and all of this to be localized to that apical domain. Now, what happens at the other domain is a collection of something that antagonizes notch signaling, and that is NUM over here. So if you segregate NUM at that posterior domain, and when you then have cell division along the axis that has been established by inscrutable, then you can make sure that all of the NUM goes into the progeny, into the basal progeny, which now can make sure that notch signaling does not occur. So if notch signaling doesn't occur, then differentiation can occur. Because if you remember, even from your lateral inhibition, where there was notch signaling, differentiation could not occur. And so you need that for self-renewal. So that's why the inhibitor of notch you need to put into your GMC. So that allows you to then differentiate. And what it also allows is this factor, which is Prospero, which is found in the neuroblast as well. But in the neuroblast, Prospero, the Miranda, which is the other thing that you saw in the movie, makes sure that Prospero does not enter. Prospero is a transcription factor, but Miranda kind of hangs on to Prospero and doesn't allow it to go into the nucleus, because if it does, which is what happens in the GMC, differentiation occurs immediately. I have a feeling I might have lost everyone. Is that true?
Yeah.
Yes, I did. Okay. Okay, let's not worry about the details. If you're interested, you can think about the details. But the important thing to remember is what all of these asymmetric things are doing is making sure you have asymmetry And in that asymmetry, you want to make sure that everything that promotes differentiation gets packed into the basal domain, and everything that ensures stem cell renewal gets packed into the apical domain. So that is all that asymmetry, that is the entire purpose of all of these complex molecular interactions. The details are not important, the factors are not important, but what is important that apically you need to make sure that differentiation doesn't occur. Basally, you need to make sure differentiation occurs. And the other thing that you need to make sure is that your cell division has to occur along this very careful segregation of differentiation and stem cell renewal segregation of factors that you have done. That's the bottom line of this slide. Okay. And I will guide you to, so this is a link to a video, that's Jürgen over there. And I will guide you to his lecture. He has 2 lectures. The first one deals with a lot of asymmetric cell, things that we spoke about now, and he summarizes it very beautifully. So do please take the time to at least listen to his first lecture. In the second lecture, he talks about how he uses human brain organoids to study these kinds of phenomena, because remember, asymmetric cell division lies at the heart of those numbers that we were talking about. So he uses organoids then to study how this very, very basic cell biological process is being used in the developing human brain. You can't do experiments with the humans, so with organoids to give you the cell numbers that you need in the developing human brain. Okay, so so shall we move on then? The summary of all of that before we move on is that the combination of asymmetric cell division and all of those different division patterns that we discussed is what come together to give rise to the appropriate cell numbers that each neural stem cell needs to give rise to. Okay, so with that, maybe let's move ahead and start talking about, so that's again, these are the things we've discussed, and now we've discussed cell division as well. And then the final thing that we're going to talk about is temporal patterning. So if you do have questions or want to talk about the cell division part of it sometime, we can do that later. But for now, let's move to temporal patterning, because it'll be nice to spend some time on that. Okay, to be able to talk about temporal patterning, let's quickly recap on spatial patterning. So at the end of all of that spatial patterning discussion that we had, we took neural stem cells that were developing, we made them molecularly distinct from one another, and we had segmentally repeating units of these stem cells along the ventral nerve cord. The brain does its own thing. It doesn't have segmentally repeating units like this, but it has 100 neural stem cells per hemi male. And at the end of this, we were able to, each one of these, again, now this is at the end of embryogenesis. So these are the larval lineages that, excuse me, that got made because of spatial patterning. And just like Atal pointed out, all of this is happening in the embryo, but then you need to go through this larval life to generate the much more complex fly, which does so many more complex things, right? It has courtship, it flies around, it has circadian biology, it shows aggression. So a much more complex brain needs to be made a second time over by the same neural stem cells through this larval life. And so the number of stem cells remain the same. You haven't added or you have subtracted some. In fact, you have subtracted some neural stem cells. You haven't added any. And you need to generate all of this much more complex brain with those many neural stem cells. And so so I kind of showed you these. So after it goes through the larval part of cell division, the neural stem cells, in the adult brain, it can now make these complex lineages that I showed you one of in the earlier slide. And of course, there isn't 3 of these. There are hundreds, right? So there's 100 per hemimeter in the brain, and there are those same ones that you've seen segmentally repeating in the ventral nerve cord. And so people have actually gone and mapped all of this. So that over there is showing you each one of those lineages in the brain, and this is just the brain, not the ventral nerve cord. Each one of these is making, through larval life, making all of those neurons. Those neurons are making complex innervations and connections through the pupal life, and at the end, these are the innervation patterns of all of those lineages in the adult brain. So the point of all of this is to say that even within these lineages, there are so many different types of neurons. You can see that heterogeneity even over here. So there's a bunch of neurons over here, but you can see that some of them seem to be innervating a whole bunch of things, and then there are some complex innervations here. Some seem to be innervating this. So there's, even within these lineages that you made different from one another through spatial patterning, there seem to be some differences within the lineages as well, and not some, actually. There's a lot of different, almost every single neuron within lineage is actually different if you go in and look at it. And that is, oops, I seem to have got something wrong. And so how do you get those differences within lineages? And this occurs via temporal patterning. So what do we mean by this? So here's one neural stem cell that's dividing over time. And you can see that the early part is, this is the embryonic division. Then there's that period of quiescence. And then there are these post-embryonic divisions. And what temporal pattern, so in the end, this one lineage would have made this one neural stem cell would have made one of those lineages that I showed you in the previous slide. But what I told you in that slide was that each one of them was different. So what needs to occur is that every time this is a type 1, you can see that type 1 division pattern over here. We're adding 2 neurons with every division. At each division, you need to add a neuron that is slightly different from the one before it. And so temporal patterning is what allows the neural stem cell to do that. So how? So we're going to talk about, there's different kinds of temporal patterning. Let's start with a typical type 1 neuroblast in the ventral nerve cord. And we're talking now only in the embryo, because the temporal patterning has been best described there. And what happens over there is that the neural stem cell, remember this is the type 1, it divides asymmetrically to give rise to 2 neurons and self-renew. Now what's happening with temporal patterning is, right at the beginning, the first thing that triggers the temporal pattern is a factor called hunchback. And when a neuroblast is in its hunchback time window, it generates 2 neurons. And through self-renewal, what occurs is the stem cell stops expressing hunchback, its first time window, and it switches to expressing a different time factor, Kruppel. And then it divides. So the Kruppel time neuroblast divides, and it makes 2 neurons and self-renews, and it shifts to a 3rd time window. And that 3rd time window is defined by PDM. And then it does that again, and it transitions to its Castor time window. And then from Castor time window, it divides again, and it goes into a Castor grainy head time window. And this kind of spans the entirety of the embryonic divisions. What the temporal patterning is doing is that each time factor, hunchback, Kruppel, PDM, Castor, grainy head, each one of these is allowing the stem cell to make a different neuronal progeny in its time window. So this has been described in bits and pieces for many lineages, but very recently it was defined for one complete lineage, completely defined for one lineage, which is called the neuroblast 7-1. And what you can see over here is that these are motor neurons. So they're all motor neurons coming from the 7-1. And you can see that this is what it looks like in the ventral nerve cord. So they kind of look similar, this one and this one. But actually, if you go in and you rotate, so this is an EM reconstruction, but if you went in and you rotated, the dendritic innervations are quite different from one another, and these are all different from these. And the most important thing about these motor neurons is that they each innervates a different muscle in the larval body wall, meaning that at each time window, a different motor neuron is being made that controls a different muscle in the body wall. And that is what temporal patterning allows you to do. Over time, generate different neurons from the same neural stem cell. I think that was a lot. Do we want to stop, pause, and digest that with some questions, discussions? Does anyone have questions?
I had a question.
Yes, Ron?
So if all of these different types of neurons arise from a single neuroblast, does that mean that there's some amount of migration involved for them to innervate different muscles?
Not migration. Actually, in Drosophila, migration is almost unheard of in the nervous system, except for glial cells. Glial cells migrate. Neurons do not migrate in Drosophila. But what it means is that from this lineage, all of these neurons that are born will form so these motor neurons the dendrites are doing slightly different things, but the axons will kind of fasciculate together and they'll all exit the ventral nerve cord. So they'll exit, I don't know if you can see anything, they'll exit the ventral nerve cord all in one bundle and then reach that segment. So even the muscles are segmentally organized, because remember the larva crawls like that, right? So it's made up of these like rings, and each one of those rings have a very organized muscle architecture, which is segmentally repeating as well. So what happens is that this lineage would fasciculate together, enter into that one segment part, and then defasciculate wherever needed to go to the appropriate muscle. So it doesn't migrate, the axons fasciculate. I don't know if you're familiar with this word. Basically, they kind of come together and they grow. It's not migration, they grow. The cell body remains where it is. They just grow until they reach the target. And then each one takes a detour, like, like off a highway. What are those called? Exits. Each one exits at different locations. It's not migration, it's growth and defasciculation. I was going to say something about you said something and I thought that I should mention. You said, do they migrate? Oh yes, that's right. So actually, let's just quickly, I don't like going back in presentations, but just for clarity. So can you see this? So that idea of fasciculation, you can see over here, Ron. So these are all neurons that are born from a single lineage. They're all slightly different. So I could show you pictures of each one of them. I don't have it here, but you'll notice that they're all growing together. This is fasciculation. All of them bundle their axons together. And then when they come here, some of them that need to innervate here will take an exit here. Some of them that need to innervate here will take an exit here. Some of them that need to innervate this nodulus over here will take an exit over here. So it's not migration, but it's growth and fasciculation, growth and defasciculation.
Okay, yeah, thanks. So they have some form of probably signaling or something to go to the right target then, right?
Absolutely. So that is a part of nervous system development that we actually unfortunately not going to cover, which is axon targeting. So a lot of work initially was done, in the 80s and 90s was a lot about how does an axon find its right target? And there are beautiful screens that were done in flies, largely flies actually, that showed you how axons navigate to find their right targets. You're right, absolutely. There have to be signals along the way.
Okay, thank you.
Yeah, any more questions?
Sonia, I had a doubt as to what this hunchback Krapfil, are they all time factors or they are factors secreted at particular points of time?
Yeah, I'm glad you asked that because I think we should talk about this a bit. So what do you think could be a time factor? What does it mean to be a time factor? Remember, we're just talking we're talking genes and we're talking proteins. So what would it mean to be a time factor? Sorry for putting the question back to you, but let's just try and riff on this a bit. So you used two words. You said it's a time factor, and then what you said, could it be, what did you say? Secrete, it's something that it secretes, you said.
Could it be certain factors produced or secreted at particular points of time that cause the stem cell to differentiate into a particular kind of neuron?
Okay, so these are actually transcription factors. So do you guys know what transcription factors are?
They bind to the. They are the ones that help in the expression of certain genes.
Yeah, exactly. So the transcription factors bind to DNA and promote or inhibit the expression of other genes. So they're very potent proteins. So these are transcription factors. The second thing I will say to answer your question is they act autonomously, meaning that they act within the cells that they're expressed in. They're not secreted and act on neighboring cells. So what you're alluding to is secretion and influencing other cells. That method is employed. We're talking about embryonic development here, embryonic timekeeping here, but in adult specific timekeeping, which occurs in the larva, there is a non-autonomous, an extrinsic cue that seems to be involved in timekeeping, and that is mediated via ecdysone actually. And that's really neat, and I can only guess at this because the larval timekeeping has to take place over 4 days. So these intrinsic mechanisms, imagine like each segment has, say, 30. Imagine, okay, it's 100 neural stem cells in the brain, each one trying to keep its own time. It's very common, because of noise in transcription translation. You can imagine how each one of them kind of lose time, right? One may be going slightly faster, one may be going slightly slower. And so over 4 days, those small changes in time differences could result in massive changes in which neurons are producing when. And in response to Ron's question, when all of these are growing and they're extending their axons, they need to find the right partners. So now imagine one lineage has somehow, for whatever reason, has made its neurons too quickly, the other one is taking more time. When these guys are trying to find trying to do pathfinding, if they don't find the right partner, that's it, connections can't be made. So it's really important to do timekeeping correctly. And because you can lose track of time over a 4-day period, these extrinsic mechanisms come in. And so there'll be this pulse of ecdysone, which anyway happens, again, beautiful reuse of signaling in biology. Anyway, larval molts happen, right? So they go from first insta to second insta to third insta. There are these spurts of ecdysone, and that somehow seems to tie into these autonomous timekeeping events to kind of synchronize them in a bit. So secreted factors do act. But for these ones that we're talking about right now, Shubha, these are all intrinsic, they're transcription factors. And actually, if you look at this slide over here, so this is from a very recent review by Cédric Morange, where he's reviewed this. So if you're interested in timekeeping in the nervous system, it's a very interesting field. We have just now spoken about this embryonic timekeeping. And notice, this is very interesting, right? Notice how in this axis of time, the embryo takes up 50% of the time, which is 1 day, and the rest of the 4 days are taken up by exactly the same amount of time. And that's a reflection of how much we know about the embryo and how little we know about what happens in terms of timekeeping in early and late larva. So in the embryo, this is why I started with that, Shubha, see, these are transcription factors. So you'll see this transcription factor, hunchback, the thing that initiates the time, turns on, activates Kruppel. Kruppel activates PDM, PDM activates Castor, and so on. So time transitions are basically windows of expression of transcription factors. But if one was just turning on the other, then you don't have neat definitions of time. right? You could have hunchback and then it's hunchback and Krupal. It's not sharp boundaries. So what ensures sharp boundaries is that as soon as hunchback turns on Krupal and Krupal levels rise, then Krupal shuts off hunchback. And then you can see that motive of, activation and repression throughout the timekeeping circuitry, genetic circuitry, timekeeping genetic circuitry. And you can see there's an additional layer of control so that you have very neatly defined times of expressions of these time factors. You ensure, hunchback ensures that you're not getting a late expression, an early expression of a late time window. So very, very intricate and complex gene regulatory networks ensure very neat definitions of transitions from one time giving factor to another. And this, again, if you're interested in computation and modeling, those kinds of things, very beautiful system, and people are actually trying to see in terms of, so how do you know that you've made that? Actually, even this is not complete, right? Because you make each lineage, even in larval life, is making 20 neurons. You don't have 20 time windows here. So people think about whether you have saturated the time, our knowledge of the time window, the temporal windows, based on the number of neurons that you need to generate. I kind of lost your question, didn't I, Shubha?
I think I've answered most of it. So in short, it's autonomous and it's something that's controlled within the cell and not by any external factors.
Yeah, but it's sometimes synchronized by external factors.
Okay.
This timekeeping is just basically transcription. That was the other point that I wanted to make. It's just a transcription factor. So then you can ask how that mediates identity. Those are interesting things that people study. What does it mean for hunchback to be making a particular identity that you one motor neuron? What does that mean? We don't fully understand that. Yes, Ayanti had a question.
Yeah. So hunchback and Krupal, there is so they come on before as well, earlier in development. Actually, I don't know whether it's before, but they do come on as I think one of those patterning genes.
Exactly.
So then I'm wondering if they do, because they do seem to be able to regulate each other. So I can imagine maybe if PDM castor is not available in the rest of the embryo, this cascade doesn't get set off. But do they regulate each other and sort of do any kind of temporal maintenance outside of the nervous system earlier on? Or do they not occur together before this?
No. So actually, I think we had a discussion about this in the last class where we were talking about some of the early anterior posterior patterning genes, and I said, they're going to come up later. And so the segment polarity genes and the segment pair role genes come up in spatial patterning. These early gap genes seem to be coming up in maintaining time. Now, your question, Ayanti, of whether hunchback Krupal do timekeeping elsewhere, I have no idea. I don't know where else during development you need to do this kind of timekeeping in Drosophila. You need to do another system where timekeeping comes in beautifully is somatogenesis in vertebrates. And that is via the mechanism there is very different. It's via oscillation of yikes. I do not remember what are the factors involved there, but it's beautiful. The factors that are involved, you have these waves and troughs of expression, which with each wave you get one specification of one somite. So Ayanti, I'm sorry, I don't know whether hunchback and Krupal do any timekeeping anywhere else, or indeed whether there's any tissue that needs this kind of timekeeping during Drosophila development. But it's an interesting thing to think about.
Yeah, and also, so they are like their pergerance, how long is this the same zygotic hunchback that does the early patterning, or is it separate?
The transcription factor is the same. The regulatory domains are very different. So people have studied the regulatory domains of hunchback, which allow for that early expression of hunchback in the embryo, that anterior domain of hunchback expression, and I think there's one slightly posterior as well. And it's a totally different regulatory domain that allows hunchback expression in stem cells. But the transcription factor itself is exactly the same. There are some, there's some interesting modulation at the level of the protein itself. So hunchback needs to dimerize for its activation role, but doesn't need to dimerize for its repressive activity. So there's some play at the level of protein there, but not in terms of the actual protein. The protein itself is the same, no matter what it does, gaping or timekeeping.
Okay, thank you.
Okay, any more questions? Okay, in that case, let's, and of course, like I said, you have my email, please. These are interesting things for me. For example, I hadn't, is timekeeping needed anywhere else? I don't know, but it's nice to think about. So we know much less. So now just, so we've spoken a little bit about how that timekeeping is kept, what it means for timekeeping. You can generate different neurons. How it does that, we understand very little about that. But that's the summary of timekeeping in the embryo, type 1 embryonic neuroblasts. And actually it's true even for type 2s in the embryo. It's the same type timekeeping genetic network. In the larva now, things are very different. And that's because it looks like that timekeeping is taken forward. So if you look after quiescence, so if you look over here, that's the period of quiescence. And the neuroblast is going from its castor grainy head window. It seems to now, after when it wakes up from quiescence, it's retained that time window. It's not like it's starting from hunchback. It's in that time domain, and then it seems to be moving into a different type of genetic network. And a lot of work has happened on this in the last few years. And as you can see, very little is understood about it. So what people have been able to show is that that entire period of 4 days is generally divided up into 2 broad domains that are specified by this early timekeeper chemo. So it used to be called chemo. Now people call it chinmo. It was always written this way, but since when I first read it, we called it chemo. I tend to call it chemo. You can call it what you like. But it stands for chronologically inappropriate morphogenesis. I think that's the name. So in the larval life, all we know is that there are these 2 broad windows specified by chemo and broad, which you see over here. And more recently, people have shown what regulates that transition from chemo and broad that you see over here. And these are some RNA binding proteins called IMP. I don't remember what IMP stands for, and SYNCRIP. And here is where Shubha, E90, can you see E93 over here? E93 is something that comes downstream of ecdysone. So somewhere over here is an ecdysone pulse, which seems to be acting to synchronize the various neuroblasts across, because remember that ecdysone pulse is throughout the whole embryo. So very little is known about timekeeping in this early or late larva. This is true for type 1 neuroblasts, but it's also true for type 2 neuroblasts. Now remember, in type 2 neuroblasts, we have to worry about 2 axes. So we have to worry about the neuroblast axis, which is going through the same type of temporal transition, of which again we know very little. So these are your same things. This is your IMP and SYNCRIP, which is being regulated by chemo, and this is regulating broad expression over here. And here's your pulse of ecdysone. So this type 2 neuroblast is, at the level of neuroblast, is doing the same temporal transitions that the type 1 is doing. But remember that the type 2 neuroblast is also generating this thing which is called the intermediate precursor. And remember that the intermediate precursor is also doing that type 1 type of division, So making GMCs in neurons and self-renewing, making GMCs in neurons and self-renewing. So there's a temporal clock there as well. And what we know, again, we don't know everything, but what we know about temporal transitions there is this. So each INP goes through at least 3 transitions. The first transition, the first factor within an IP is dikeet. It turns on something grainy head, which you can see is actually also a factor in the neuroblast. And grainy head then turns on isless, and there are these back repressive motifs in that ensure these are clean transitions. And then in the INPs, so remember there are only 8 neuroblasts that go through this in each half of the brain, right? So in INPs, the late events, so in terms of what neurons they give rise to, we know that the late born cells are actually glia. So I don't know why it's being shown over here. But okay, maybe they're showing glia here. The late born ones are glia, and the early born ones are typically neurons. And for 2 INPs, these are complex experiments to do. But the same group that first identified chemo actually went in and mapped each 2 INPs from one neuroblast and showed the individual neurons that are born from within one INP. And so for that, I'm not showing you that image. I think I couldn't dig it out last night. But basically for 2 INPs, remember we're talking about so many, and these are difficult experiments to do. But for 2 INPs, what it means to have this temporal transition of dikeet, grainy head, isless, people have shown for 2 of them what it means in terms of neuronal identity. And these type 2 neuroblasts make up a structure in the brain called the central complex. So these are really beautiful, beautiful neurons. So if I can dig up that picture, I will put it in into the slides for your Moodle when this is uploaded over there. Okay, so and then something that we did not speak about at all was the optic lobe. Now, the reason we didn't speak about this was because the rules in the optic lobe for many of the things that we've discussed so far are slightly different. So even how a neural stem cell is specified in the optic lobe is very different from what we have discussed. But despite that, so there's a lot of symmetric division to make sure you get enough number of neural stem cells. The selection is not via these proneural clusters in this way, but it occurs serially. So it's those early events are slightly different. And studying the optic lobe is a more recent phenomena. I think maybe 10 years at most. People, I wouldn't even say that, 7 years people have started looking at the optic lobe, and actually they've made beautiful headway. So actually, if you're interested in this kind of thing, very rapidly in these 5-7 years, people have shown exactly the same things. So despite those mechanisms being different, they've shown that you first need to set out spatial patterning. So very quickly after the neural stem cells are specified, there are these regionalization with spatial transcription factors. And those spatial transcription factors are totally different from your anterior, posterior, dorsoventral patterning genes. So not the same guys at all. And then after that, you need to do temporal patterning, like we've discussed for the rest of the nervous system. But again, it's not the same factors. So you'll see over here, it's homothorax, clumpus, isless. So I guess some factors are conserved. Sloppy paired, dikeet, okay, another one conserved, tailless. So that's the temporal transition that happens in the medulla, the optic lobe neuroblast. But the reason I put this up, it's there in this review if you're interested. But the reason I put it up was, everything that we've discussed, you make the neural stem cells, you give it positional information, then you do temporal patterning. Look at this system that does it totally different, but the concept is the same. The genes are different, the concept is the same. And you will see this in Bhavna's lectures as well. You're talking about a vertebrate over there, but the concept will be the same, where you have to do spatial patterning, you have to do temporal patterning to give rise to all of those different types of neurons that you're interested in. Okay, so we spoke a little bit about what it means to do temporal patterning and the neurons that it's giving rise to. And here, like I was saying, these are not easy experiments to do. They take a lot of time, very complicated genetics. But in the fly, people have been able to do it for some. I showed you an example in the embryo with the motor neurons innervating the different muscles. I referred to the one in the central complex where the INP lineages for two INPs have been shown. I'll put up a picture of that. And here's a type 1 neuroblast where actually these guys, so the same gang that described chemo, actually took one neuroblast that makes up one of the antenna. Actually, I showed it to you in one of the previous slides. It makes up the olfactory neurons for the olfactory circuit, and they've gone in and mapped every single neuron that comes out of it. So this is the one neuroblast where we know everything. We know every neuron that's born out of it. And just to illustrate what that means, so here are names of the neurons. They call these funny names because these are the names of glomeruli. Now, the olfactory lobe or the antennal lobe in flies is like a bunch of grapes. And each grape in that bunch of grapes corresponds to a structure called a glomerulus. And that glomerulus it's not just a grape, of course. It's taking in information from the antennae of the fly, and each glomerulus gets one class of olfactory receptor bearing neuron. So that means that if all of those neurons coming to one glomerulus have one receptor, that means they have a similar odor profile that it senses. So functionally very different. Each glomerulus is functionally very different because it receives information about unique odors. And the projection neurons, which you're looking at over here, are then innervating that particular glomerulus. So there are different projection neurons that each go to individual glomeruli. Their dendrites are there, and then they take the information up to higher brain centers in the mushroom body, a place of learning in the fly brain, and the lateral horn. So that's a lot of information, I know, but what we're looking at is a lineage that makes those projection neurons. And what you're looking at are different types of projection neurons, and they're called, their names, oops, what did I do? Their names, they get their names from the glomeruli that they innervate. So we're going to pay attention to the magenta neuron. So you can see this magenta neuron is sitting over there, and it's innervating this VA4 glomerulus. And they've shown you just a repeat. So this is the second one that's going to the VA4 glomerulus. This projection neuron, born a little bit later, innervates VC2. You can see another one that does the same. This one innervates VC1, another one that does the same, and so on. They've gone on and shown this for very many projection neurons. And correspondingly, they're showing what the axonal projection looks like in the lateral horn. You can look at this later, and they're showing only one over here, but basically you can see that even in the lateral horn, the projection pattern of these projection neurons are different. And they've summarized all of this. This is a lot, but do look at it whenever you can. But they've summarized this in this beautiful table over here. And what it shows you, okay, let me explain. So these are the names of the various projection neurons that are born from that one lineage. And this is the time at which that projection neuron is born after larval hatching. And so you can see how they've ordered it such that these guys, these projection neurons, the UNPN8 projection neurons are born very, very at the very end, whereas these guys, these lavish DA1 neurons are born somewhere in the middle. So you can see how each projection neuron, very carefully mapped, is being born at a particular time point. Now remember that all of this has to be specified by your temporal transcription factors. And look at the number of time windows you need to be able to get all of this, and look at what we know about temporal patterning in the post-embryonic life. So lots to do still. But that's the connection I wanted to make, that your temporal patterning events have to make all of these different neurons within lineages. Okay, so we're coming to the close of the class, a final comment. So now we're going back to the embryo where we're talking about the the earliest timekeeper, hunchback. So here's a picture I showed you of one of the lineages in the brain. This in the ventral nerve cord. This is the neuroblast 5/6, and that on the side over there, these two neurons are the neurons that are born from neuroblast 5/6 in the hunchback window. So we know that, we've mapped that. We know that neuroblast 5/6 makes those neurons, which we decided to call chase lounge, because it looks like somebody chilling out on a chase lounge. And then this neuroblast over here, which makes a totally different lineage, out of that same time window, that is the hunchback time window, is making a neuron that looks very different from the chase lounge. This is called the G neuron. So it got the G neuron name because actually, so everything that I've told you so far in terms of the neuroblast was actually first discovered in grasshoppers. So neuroblast mapping with all of those numbers, 1, 2, 7, 4, 5, 6, come from grasshopper names. And it's only when people realize that you can't do genetics, when the molecular biology revolution took place in the 80s, people realized you can't do genetics in grasshoppers. So then there was this huge movement to map all of this in Drosophila so that you could then use Drosophila to do all of the genetics to understand the molecular basis of all of this. So the G neuron, the hunchback-born G neuron of the 7/4 lineage, is called that because of its grasshopper counterpart. But the thing I want you to notice in this slide is that two spatially distinct neuroblasts, 5/6 and 7/4, make two different neurons, chase lounge and G, in the same time window. So hunchback, as a transcription factor, is specifying two different neuronal identities in these two stem cells. See, Shubha, this is what I meant. What does it mean for a transcription factor to specify neuronal identity? Same transcription factor, different spatial neuroblast, different neuronal identity. And so the idea is that somehow a neural stem cell must integrate what it is. It must know its spatial identity and then utilize its timekeeping to make an appropriate neuron that is appropriate to its spatial unique molecular identity. And so that's the bottom line from this class. We're 2 minutes away from the end. A stem cell needs to integrate spatial information with its time information, where in the time of development it is, where in the axis of development it is, and then decide, I use that word, what is the appropriate neuron to make? Is it that, what was the name, DANDI-DA1 neuron, or is it the chase lounge neuron? That decision comes from integrating spatial information with temporal information. And all of this comes together to generate all of those different types of neurons that we spoke about in the previous class. Okay, so it's 10:59, and I'm going to leave you with this picture of the Drosophila, a small subset of the lineages in the Drosophila embryo. And I will be very happy to take any questions now or later. Okay, and any questions, guys? Questions, comments? I think this one was a bit overloaded, right? Got a bit heavy towards the end. Okay.
Yeah.
Girish said, yeah.
I had one.
Sorry to bombard you, Girish.
It's okay. I had a question about the asymmetric division in cells for the neuroblast.
Yeah.
Is there an animal which does not do that? I mean, if it symmetrically divides, you get 2 to the power something cells, which is much faster than asymmetric cell division.
Oh, I see what you're saying. I see what you're saying. So symmetric division occurs, and so if you have symmetric division and you make sure that all you're doing is ensuring that differentiation doesn't occur. So you need to do both of those things. If you do symmetric division and ensure that differentiation doesn't occur, then you're expanding your progenitor pool. This occurs in the in the optic lobe in Drosophila development. It occurs a lot in the vertebrate. You'll hear from Bhavna's lecture. To ensure that you expand your progenitor pool, very definitely symmetric cell division while ensuring that differentiation doesn't occur is a strategy used in many organisms, many tissues to ensure you expand your progenitor pool. Yeah, very much.
I see. Yeah, thank you.
Any more questions? Okay, in that case, Varsha, what instructions for me? Sorry. Is that my internet connection or?
I can't hear her either.
Okay, okay.
Varsha, breaking up.
Varsha, your voice is going into electronic mode. Into wah-wah voice. Okay, I'm going to assume you're saying that the class is over. I will also, there's a comment, 1 second. Okay, it just says, hey. Who will be teaching next class? So Bhavna is teaching next class. She's going to cover, is Bhavna?
Yes, hi.
Bhavna's here. Oh gosh, you were all quiet. Bhavna, you should have interrupted and filled in with the vertebrate bits.
Oh, okay. I didn't want to interrupt the
No,
class.
if I was saying something
No.
dramatically wrong or whatever.
So, hi everyone. I'll be teaching the next class, and I'll take on from where Sonia left, like taking you into how vertebrate nervous system development is different.
Yeah, and I'll be around for that as well because there's a lot that I have to learn about vertebrate development as well. Okay, guys, thank you all very much, and see you all on Friday. And like I said, if there are any questions,